Welcome back. We are starting with chapter 7, which is bacterial growth and archaeal growth. So we are going to talk about the most of the bacteria and the archaea that they produce by the binary fission. We are going to first take a look at how they reproduce, just some basic strategies of the cell division, and then we will go into the general mode of archaeal growth, and then we will going to see the growth patterns and the other things. So the first thing, this figure shows that the steps in the binary fission with a young cell at early phase of the cycle. Next, parent cell prepares for division, enlarging its cell wall. Now the cell membrane, overall volume, everything is going to be changed. Now the DNA replication starts right here. And next, what happens that once the DNA is being replicated, the two cells are going to get separated. And you can see that the chromosome is distributed in each of the cell. And the cell is a little bit smaller than the original cell from where it started the division, so they will grow in size a little bit as well. So the most important part of the thing that before the cell separates into two individual cells, the DNA need to be replicated and segregated towards the pole of each cell before the division of the cell happens. And the process is a binary fission in bacterial cells as well as archaeal cells. So top left, you are seeing that this is a budding Listeria monocytogenes mother cell, and the bud is being formed. On the other hand, here you can see that these are the biocytes produced by this cyanobacterium, and the cyanobacterium is named as Dharmacocarpella, and individual cells are being formed, multiple individual cells, and they will come out from these cells. On the other hand, here in the bottom, you are seeing that the spore formation by the Streptomyces species, and the illustration shows a spore. Then you see there is a germ tube, vegetative hypha, branch, and the aerial hypha, and all these different structures. And then the formation of the spore at the very top of this aerial hypha. On the other hand, you can see here another picture of the same thing, that this is a schematic, this is a real picture of this Streptomyces species, which is a filamentous bacteria. So next we are going to talk about the bacterial cell cycles, and which can be divided into three phases. So the cell cycle, the sequence of event, the three phases are, the first we are going to look at after the cell is born. We're going to see the chromosome replication and partitioning and the cytokinesis. So most of the bacteria do have a single chromosome and also a single origin of replication that we have talked about in the previous chapter. And the site which replication begins and the replication fork from the replication origin, there will be two forks and each will move towards the terminus. Terminus is a site where the replication is going to get terminated, and this is located usually, if the chromosome is circular, it's at the opposite pole. The replisome, we also talked about that in the previous chapter, are the group of proteins needed for the DNA synthesis, and this replisome starts the synthesis of the DNA at the replication fork, where it gathers all these proteins for the machinery for the DNA replication. And again, DNA replication proceeds in both directions from the origin, as I've said, and origin moves to the opposite end of the cell towards the terminus. So let's take a look at this. So here, the initiation mass reached means that the cell has completed its vegetative growth. What do you mean by the vegetative growth? The cell is completed its gradual growth. It is grown up. Now it can divide into two. So this results from an increase in the mass of the cell, which also result in the accumulation of a particular protein, which is known as a DNA A protein that we talked about, which initiates the DNA replication. Right here, the initiation of replication happens, and this initiation of the replication, the cell is becoming ready for the DNA replication. The origin of replication migrates at the center of the cell, and protein that make up the replisome assemble around the origin as well. So as DNA replication proceeds, the newly synthesized chromosomes move towards the poles, and the cell increases in length as well. So here, the threshold cell length is reached, and the cell reaches a certain length, which is known as the threshold length. And here, what happens that gradually there will be an initiation of septum formation. The initiation of septum formation is triggered by the cytokinesis, produce two daughter cells that have inherited one chromosome each. And in this illustration, what you're seeing that this is one round of DNA replication is completed before the cell divides into two cells. And rapidly growing culture, say for an example, for E. coli, this generation time for this cycle is about 20 minutes. And the second and the third round of DNA replications are initiated before division of the original cell is actually completed. Thus, the daughter cells inherit partially two replicated DNAs. So this is basically the cell cycle in the E. coli. So here we are looking at the mechanism by which the chromosomes are partitions to each daughter cell. Has not been fully elucidated, but studies of the Colobacter crescentus have provided the most complete understanding for us for this partitioning process. So this par A and the par B, these are the key players, and they work together to initiate this partitioning. So the par A is able to polymerize to form these filaments, and par B is a DNA binding protein that binds a site on the chromosome called as a par S, shown in green. Now the par B binds each par A site on the two daughter chromosomes. So you can see that there is right here, this is a par S site, this is a par S site, and to each of them you have one chromosome right here, another chromosome right here, it binds to both of them. Now the par A is a cytoskeletal protein that pulls one of the daughter chromosome towards one of the pole up to the opposite of the stalk pole. So this is an interesting bacteria where the bacteria is attached with a stalk. And we're not going to go into the detail, but this bacteria will lose that stalk and it will be replaced by some of the flagella. They are called the flagellar swarms, and these bacteria will spread and then they again will attach to some substrate with that stalk, and this goes on. So this is towards that stalk. That's the only thing that we are pointing out here. So what happens? The par A pulls the chromosome, which is being separated, towards the pole. And the other proteins that are involved for localization of the par A and the par B, there are many other different proteins are there. So basically what happens that in this region, the whole chromosome is covered by the par B protein initially. But this is depolymerized as it is taken over by the par A and which is going towards the pole. So remember that these par B covered this area as well, but as it is being moved towards the par A is coating this, separating this strand, and initially it was covered by the par B that get depolymerized. So this is the process of pulling the two chromosomes towards the pole of the cell. So here we are looking at the cytokinesis, and this is the formation of the cross wall between the two daughter cells. And there are several steps: selection of the site for the septum formation, and assembly of the Z ring that is composed by a protein which is a cytoskeleton homolog, which is known as FTSZ. And we are going to look at the assembly of the cell wall synthesizing machinery close to the FTSZ and constriction of the cell and the septum formation. So here the correct separation actually required that the Z ring is formed at the proper place along the center line at the proper time. Now, in E. coli, there are several proteins which are called a mean CDE system. So the proteins are mean C, mean D, and mean E. These actually limit the Z ring formation inside the cell. Now, why it does that and how it does that? These mean C, mean D, and mean E, these proteins oscillate towards the end of the pole of the cell. So what happens, say for example, this is being shown that this protein is being polymerized in one cells. So that prevents the formation of Z ring in the center. As it is being depolymerized, it is moving away from the center On the other hand, as it moves away from the center towards this end, again, these proteins are going to get polymerized towards the other center, and that is again pulling this towards this center. So oscillation in between that each and every pole moves the formation of the Z-ring formation in the middle of the cell not to happen. And why this is important? This is important because what happens before the chromosome gets separated into each of these compartments, if by chance the constriction of the Z-ring happens, it will slice the chromosome off as it is migrating towards the pole. So this oscillation moves this ring back and forth constantly so that the septum formation cannot happen in the middle portion of the cell until there is a signal and until there is a time, which is basically the separation of the true chromosome into each side of the cell. So the cell division apparatus is composed of numerous proteins, and the first step in the divisome, which is divides the cell, is the FTS-Z polymerization to form the ring. And how this ring formation happens, this happens towards the cross section of the cell. So if this is the cell, it doesn't happen lengthwise, but it happens around the cell, around the cross section of the cell. And this is called a process which is called an FTS-Z treadmilling. As FTS-Z is being polymerized from here, one end is depolymerizing, which is adding the depolymerized subunits to the other polymerizing unit. And as this happens, this also shortens, and in the shortening process will make the septum come together and make the septum invaginate to inside the cell, and that will constrict the plasma membrane and the peptidoglycan to separate them in two different chambers. So here you can see that along with the FTS-Z, there are some other proteins which are called FTS-A, and these attach the plasma membrane and the FTS-Z. So this is essential so that the plasma membrane is constricted as it is being pulled by the FTS-Z. And along with that, these are the peptidoglycan synthesizing enzymes, which will synthesize the peptidoglycan as it is being constricted as well. So this is the process, how the divisome forms and how the cell is going to produce the Z-ring to separate them. So here are some of the players that we have seen. FTS-Z forms the Z-ring, positions the peptidoglycan synthesis enzyme for the septum growth, and FTS-A and the ZIP-A anchor the Z-ring to the plasma membrane. As you can see that it's Z-ring right here. This will form the ring and anchors to the plasma membrane. Then if you have an FTS-K, this helps in the chromosome segregation, separation of the chromosome dimers. That is essential. And then you have an FTS-EX, coordinate the hydrolase of the septum growth and the cell separation. Here also another FTS-I and FTS-W. This FTS-I is a penicillin binding protein. We're going to talk about that later. And this helps in the peptidoglycan synthesis in the septum. And then you have an FTS-N, FTS-QLB. These coordinate the ring constriction with the septum growth. Now, you do not individually need to go in every one of them and their functions, but just have an overall idea that what are the proteins involved in the divisome. So here what we are looking at is the peptidoglycan synthesis, which starts in the cytoplasm with the attachment of a uridine diphosphate to the sugar in acetylglucosamine, which is known as a NAG. Some of these UDP NAG molecules are going to convert it into the UDP NAM, which is an N-acetylmuramic acid. And then these are going to get attached with a bacterioprenol. And these are lipids or membrane spanning lipids. And once these NAG or NAM is attached to them, they are going to flip. What it means by that, this end become this end becomes this end. This is an energy requiring process, and this will need input of energy. And once they are flipped on the other side, you can see that the NAG and the NAM is now up towards the exoplasmic phase. And here there is some protein which is known as a autolysin, and you can see that they are designated as A's. So these autolysins are going to lyse the peptidoglycan or break the peptidoglycan in certain areas so that the new NAG and NAM can be inserted into those areas so that the cell wall growth continues. And that's the function. So you break up the existing peptidoglycan and then you insert new new NAG and NAM, which makes the peptidoglycan, and you resynthesize also the bridge, the peptide pentapeptide bridges. So this helps the growth of the cell wall. Now here we are looking at the cell wall growth and the cell shape right here. And the FTS-Z determines the site of the cell wall growth. And if this is a bacteria which is a spherical, you can see that it is always towards the medial area. And this is the area of the cell wall growth. You can see that once they separate, this is the area where the cell wall is growing. But what happens, the rods are similar but elongate prior to the separation. So what you are seeing in this picture, that the cell wall growth is happening all throughout. So this is where the cell is really growing in size from smaller to the larger. Once that threshold is reached, means the cell is okay to divide and the DNA is going to get replicated or the chromosome is going to get replicated. At that time, the FTS-Z prepares the cell so that the cell wall growth happens only around the medial center, not randomly all throughout. So here you can see that it is happening towards the medial center, and this is the process by which the cell wall grows. And also what happens, that the MREB protein is actually very important, which is also a homolog of a cytoskeletal proteins. So MREB determines the cell diameter and the elongation ratio, and that is also very important for the growth of the cell wall. So here, on the other hand, Vibrio is a comma-shaped cell, and here also you have the involvement of MREB and the FTS-Z as well. So this is a helical polymerization throughout the cell. But the interesting part is that there is also another protein which is known as a crescentin, and this was studied in Caulobacter crescentus, or the Vibrio cholera do have the same structure as well. And this is the crescentin, which is also a homolog of cytoskeletal protein. And this grows in one side and it localizes to this short carved side of the cell. And that's the reason that the cell divides in an asymmetric shape. The cell is going to divide more on this side, less on this side, and it takes a comma-shaped signature structure of the Vibrios like this Caulobacter. So this is the role of crescentin, to have an asymmetric growth to give rise to a comma-shaped bacteria. So now we are going to take a look at the archaeal cell cycle, and we're going to look at one of these, which is known as very well, is the sulfolobus. And we're going to compare that why they are similar or different from the bacterial cell growth. So here indicate that there's a cell cycle which is similar to the mitotic cycle in the eukaryotes. So you have a G1 growth phase, and once that growth phase stops, there will be a DNA replication, the archaeal DNA replication, which we compare with the S phase or the synthetic phase of the eukaryotic cells. Then you have a G2 phase, which is a second growth where the cell prepares to separate the excess amount of DNA or separate the chromosome. Finally, the segregation of the chromosome, which is basically the mitosis from prophase to the telophase, followed by the cytokinesis. It is similar. It is not exactly the same, but it's similar to the eukaryotes. So the segregation occurs using a seg-A and the seg-B proteins. This is the segregation of the chromosome. And that is similar to the bacterial partitioning system. If you remember that we already talked about that the par A protein binds to the one of the chromosome, which is pulling it towards one end, whereas the par B binds to the other one. So this seg A and B are similar to the par A. Seg B is unique to the archaea, but the function is similar to par B as well. So you have some homologous functions for that. But the DNA sequence is similar to par S, which coordinate with the par B protein. That has not been yet discovered in the archaea, and we are still learning about this process. Now, on the other hand, the archaeal cytokinesis is similar to eukaryotic cytokinesis. Rather, we will say that it is more similar that how multivesicular buds actually form from the endosome. So let's take a look at what I mean by that. So they use a protein similar to these escort proteins. So what are the escort proteins? Escort protein stands for endosomal sorting complexes required for transport. These proteins are essential for the eukaryotes. So what they do? These are responsible for transferring proteins from the endosome into the plasma membrane or to be secreted outside. So they assemble in the endosome and form a vesicle, and these vesicles compartmentalize specific proteins that need to go towards the plasma membrane. So escort proteins take them to the plasma membrane so that they fuse, and that's basically the process. But this is also important in the eukaryotes, that the pinching off of the two cells or from the neck that is being attached to each one of them is also used by this escort protein. And there are 4 distinct complexes involving forming multivesicular bodies, or MVB. The complex escort protein called escort 3 circles the neck that I was talking about like a spring, forming an invaginating membrane and brings about the membrane scission. Scission means that it is just cutting the membrane so that two actually separate. And there are 3 division proteins that have been identified in sulfolobus, which is this CDVA, escort3, and that VSS4. And this is very, very different from the bacteria. In the bacteria, this doesn't have FTSZ. The Z-ring formation is the way that the bacterial cell divide, or the cytokinesis, similar process of cytokinesis happen. But here in the archaea, this process is more advanced, more like an eukaryotic system. So in the next section, we are going to consider the growth curve, and it consists of 5 phases. We are going to describe the hypothesis, correlate changes in the nutrient concentration, and relate them to the generation time. And these are some of the things that we are going to consider. So growth is an increase in cellular constituent that may result in the increase in the cell number or increase in the cell size. So the growth refers to the population growth rather than the growth of an individual cell in the bacteria. So the 5 phases of growth curve are identified on the growth curve. You can see that there is a lag phase when organisms are introduced into a fresh culture medium, but there is no immediate increase in the cell number occurs because they are still getting adjusted with the media. And after that, there is an exponential phase, log phase, where the microorganisms are growing and dividing at maximal rate possible, given that there is genetic potential. Then there is a stationary phase where the population growth ceases and the growth curve becomes horizontal, because at that time, most of the time, the nutrition depletion is one of the cause. And after that, you have a death phase. In the death phase is opposite to the exponential phase of growth, where the bacteria are exponentially dying. And the reason for that, one is the depletion of the media, and another one is important, which is the toxic metabolite buildup. And then after that, there are it becomes a little bit horizontal. It doesn't really go all the way down here, and it just comes up to some extent and just becomes horizontal. And this is known as a long-term stationary phase. And during this time, the bacterial population continually evolve so that the actively reproducing cells are those best able to use the nutrients released by their dying and best able to tolerate those accumulated toxin. So this phase is marked by a successive waves of genetically distinct variants, which is being shown by these different curves. So there are a small number of bacteria that can stay in there, which are viable under these harsh conditions. So the lag phase, let's take a look at that. The cell synthesize new components, for example, replenish spent material, for example, adapt to a new media, other conditions, varies length, in some cases can be very short or even absent. On the other hand, the exponential phase, also called the log phase, because the rate of the growth is constant maximal, and the population is most uniform terms of chemical and physical properties during the phase. And this is the phase where you target your antibiotic, because as they are actively synthesizing everything in the cell, it is easy for the antibiotic to target those enzymes which are responsible for their replication, so that the growth can be stopped. So the most of the time, the antibiotics works best in this exponential or log phase. The next is the effect of the changes in the limiting nutrient concentration on the total microbial yield. So the chart shows a straight line of yield to nutrient concentration. However, at a sufficient concentration, the total growth will plateau. On the other hand, here, the effect of the growth rate is lowest at the nutrient concentration, but eventually it plateaus. No increase in the nutrient concentration will make it rise beyond that growth rate. So these are two examples of that. The abundance of nutrients allow the transport mechanism to be saturated, so the growth increase is minimal for adding more nutrients. So what do we mean by this? Suppose you have a specific number of glucose in the media. You are increasing the glucose concentration into the cell, but the population and each cell do have a specific number of glucose transporter onto its plasma membrane. It cannot be more than that. So in that population, no matter what, even if you increase the amount of glucose, once it reaches the bacteria or the population's maximum capability of intaking glucose, it will plateau off. So not necessarily that when you increase the concentration, it will go in this direction forever. It will plateau out just like an enzymatic saturation curve. So the stationary phase, the closed system population, growth eventually ceases. What do you mean by a closed system population? Means they are growing in the test tube, they are growing in a flask, something like that, but usually not in the environment where there is a constant supply of the nutrients. Closed system means the supply of the nutrients is not forever. And the total number of viable cells remain constant, and the active cells stop producing, or reproduction rate is balanced by the death rate, and the population may cease to divide but remain metabolically active. And as I said, the possible reasons: nutrient limitations or depletion, limited oxygen availability due to the dense population of the growth, toxic waste accumulation, and it started to do that. And also the critical population density reached means every different bacteria do have a specific different critical population density. Beyond that, they cannot live. On the other hand, the death phase is a number of viable cells decline exponentially, with the cells dying at a constant rate. Now, the detrimental environmental changes such as nutrient deprivation, the buildup of toxic waste cause irreparable harm to the cell, and they will die. So on the other hand, the long-term stationary phase, the bacterial population continuously evolved, and the process is marked by successive waves of genetically distinct variants. We have seen that. And here, the natural selection occurs. Means after a huge amount of toxic metabolite, some bacteria may be selected, or some of the strains of the same bacteria is selected, which is much more tolerant to these toxins. So here we are looking at the mathematics of the growth, and we will really not go into the detail, and we are not going to ask any of these math questions, just some basic consideration that generation time is the time required for the population to double in the size. And this varies depending upon the microorganism in the environmental conditions. Some of the bacteria like E. coli can divide 1 from 2 in 20 minutes, whereas, say for an example, a bacteria like a Mycobacterium tuberculosis, which causes a TB, the growth time is similar to an animal cells. So it will divide in 20 to 24 hours from 1 to 2 cell. So the generation time varies significant. So you can see that some of the bacteria, it could be even days. So here we are looking at that the time 0, 20, 40, 60. And if the generation time is 20 minutes, you can see that this is the number of cells that you're going, and the growth is exponential. That's what we mean by the exponential 0, 1, 2, 3, 4 to the power n. That's the growth. So here we are looking at the calculation of the growth rate constant. And again, we are not going to go into the mathematics, just to have an overview of that what it is. The N0 is the initial population. NT is the population at time t, and N is the number of generations in time. So from here, you can find out this, that NT, the population at time t, is N0, which is the initial population, multiplied by 2 to the power N. And that's the formula to calculate the population that is reproducing by binary fission. And then from there, if you solve this, what you get finally, you're calculating the growth rate constant. So what is that growth rate constant? Finally, you can get that k equal to 1 by g after these substituting this. The generation time is the reciprocal of the growth rate constant. So this is the important thing that we derive from here, that g equal to 1 over k, which is the growth rate constant. So the generation time is reciprocal to the growth rate constant. So that's the overall take-home message from the growth rate and the generation time. So here we are looking at the lag phase, and here we are looking at the exponential growth phase. You can see that how this scale is in a log scale, and that is all being shown here. So here, there are some of these bacteria, like an E. coli. If it's incubated at about 40 degrees Celsius, you can see that the generation time is 0.35 hours, which is equivalent to approximately 20 minutes. So you can see the Staphylococcus aureus is about 0.47. See, for Clostridium botulinum, it's 0.58, which is very close to about 1 hour. On the other hand, Mycobacterium tuberculosis is approximately 12 hours is the generation time. Treponema pallidum, 33 hours. Some of the Archaea, even some of them are 45 hours. You can easily see that. Some of the protists or the fungi are much more. So these are the general idea. You do not have to memorize anything, that it varies a lot. So next we are looking at the environmental factors that affect the microbial growth. So let's take a look at these. So the influence of the environmental factors are very important for the microbial growth, and some of the bacteria grow in the moderate environmental conditions, but we also know that there are many which are called the extremophiles and grow under harsh conditions that will kill most other organisms. So let's first take a look at the solute, osmosis, and the water activity. So in this part, what we are trying to understand is the role of water in the environment of the cell or the bacterial cell, that how it affects. So the changes in the osmotic concentration in the environmental of the microbial cells. So if you put the bacteria in a hypotonic solution, which is a lower salt concentration, the waters enter the cells, cells swell and burst. Now remember that this condition is a hypothetical condition. This will say, for example, if you take the red blood cells and place it in the distilled water, which is a hypotonic solution, this is the case that will happen. But for most of the bacteria, this is not the case. The reason that the bacteria do have a rigid cell wall, and that's the reason that it will prevent the bacterial cell to swell and burst. On the other hand, this is also another important thing, very important, that to transport water inside the cell, the water cannot diffuse inside the cell through the plasma membrane. It needs some carriers known as aquaporins, and these aquaporins are the channels through which water can cross the plasma membrane to go inside the cell. So if there is a bacteria where on the plasma membrane there is no aquaporin, or there is very little amount of aquaporin, then the water from outside is not going to enter into the cell to cause any harm either. So these are some of the real life considerations that may happen. On the other hand, this hypertonic solution is a higher solute concentration, and in this concentration, water leaves the cell and the membrane shrinkage from the cell wall may actually occur. This is actually more common in the nature because many times the water is actually released from the cell by at least some of the aquaporins that is present on the plasma membrane of different bacteria. So this is actually the way or the principle behind the pickling processing. So think about that. We are using the vinegar or the brine, and this makes that solution hypertonic to the bacteria. So at that time, water leaves the cell, membrane shrinkage happens, and that will stunt the growth of the bacteria. So not necessarily will kill the bacteria, but will help, but they will not allow them to divide and grow either. So this is the reason that hypertonicity stops the growth of the bacteria. So microbes also can adapt to the changes in the osmotic concentration. So reduce osmotic concentration of cytoplasm in the hypotonic solution. And there are mechanosensitive channels in the plasma membrane allow the solutes to leave actually. So this is another way that the bacteria can fight the environment. And also increase the internal solute concentration with compatible solutes to increase the internal osmotic concentration in hypertonic solutions. These are all different ways that if you place the cell or a bacterial cell into hypotonic or hypertonic solution, there are ways that bacteria can fight or adapt themselves to the change in the environment. And these solutes are compatible with metabolism and growth, and that has to happen as well. So here is an extreme example of the adapted microbes, and these are known as the halophiles. Means that these are very, very salt tolerant, grow optimally in the presence of sodium chloride or other salts at about concentration above 0.2 molar. And some of the extreme halophiles also can require salt concentration up to 3 molar to 6.2 molar. These are found in some of the salt mines as well. An extremely high concentration of potassium is also required, and the cell wall proteins, plasma membrane require high salt to maintain the stability and the activity. And you can easily see that the different groups that is being named. Right here, you have the sodium chloride concentration from 0 to 1, 2, 3, 4 molar, and this is the growth. So non-halophiles, you can see that as you are increasing the concentration of the salt towards 1 molar, the growth immediately drops. Whereas see that moderate halophile, say about 0.5 molar to approximately 3.5 molar, whereas a halotolerant can grow all the way up to about 3 molar, whereas the extreme halophiles can start from 0.2 molar and optimal can go beyond go up to the 6.2 molar. So even at 4 molar, it is actually attaining the highest growth rate. So these are some of the extremely adaptive microbes which are known as the halophiles. So here we are going to consider the solutes and the water activity, and what we mean by the water activity, which is denoted by AW. The water activity of a solution is 100 the relative humidity of the solution. So this is also equal to the solution's vapor pressure, or this is P solution, to that of the pure water. So again, the ratio of the vapor pressure to that of pure water. So low water activity means that most of the water is bound, not available to the microorganism. And most organisms only grow well at water activities around, say, 0.98 or higher. On the other hand, the osmotolerant microbes can grow over wide ranges of water activity, but optimally at higher levels. So what we mean by the available water or the water activity, just as simple as you are adding salt, just for an example, table salt or sodium chloride in a cup of water. As you are adding the salt into just pure water, the amount of water activity is going down. So most of this water is now bound or attaining this solubility state of the sodium chloride, and is not available for the bacteria to grow in there. So as the water activity goes down, as you are attaining the higher sodium chloride concentration, just like here, the water availability or water activity is going down. So this is great for the halophiles. For most of the bacteria, you can see as you decrease the available water, the growth rate will significantly suffer. So next one, which is important, is the pH and measure of the relative acidity of the solution. And this is considered as the number of hydrogen ions or proton that is present in a particular solution. And this is basically the negative logarithm of the hydrogen ion concentration. So what we mean by that, if you have higher hydrogen ion or the protons in a solution, it's going to be pH less than 7. As you're H plus going down in number, you will have a higher pH. So it's a reciprocal relationship. Remember that. It's a negative logarithm. So acidophiles are the group of bacteria which grow at optimum temperature in between 0 to 5.5. Neutrophils going in between 5.5 and 8, whereas the alkalophiles, also known as alkaliphiles, grow in between 8 to 11.5. So a wide range of growth characteristic of different microorganisms. So here at low acidic end of the scale are a human stomach fluid, lemon juice, acid. These are some of the different types of material where some of the bacteria can grow. On the other hand, say the milk, water, human blood are neutral around pH, and they say household ammonia, bleach, oven cleaners, these are alkaline end of the scale. So many archaea are acidophiles as well, along with the bacteria. For example, the Archaeum sulfolobus acidocaladrius is a common inhabitant of the acidic hot springs. It grows well from pH 1 to 3 at high temperature. And then another archaea, which is a ferroplasma, can actually grow very close to pH 0. And there are alkaliphiles as well, are distributed among the 3 domains of life, and they include the bacteria belonging to the genera Bacillus, Micrococcus, Pseudomonas, Streptomyces, yeast, filamentous fungi, and numerous archaea. As the seawater is a pH about 8.3, all marine microorganisms are alkaliphilic or alkalophilic. So the microorganisms respond to the external pH chain using the mechanism to maintain a neutral cytoplasmic pH. And the neutrophiles exchange the potassium for the protons. And on the other hand, the acidic tolerance response pump protons out of the cell. And some synthesize acid heat shock proteins that protect these proteins from denaturing. And many microorganisms change the pH of their habitat by producing acidic and basic waste products. Classic example of this is the Helicobacter pylori. Helicobacter pylori is one of the causal organisms for gastric ulcer and sometimes gastric cancer. They inhabit into the wall mucosa of the stomach where the pH is close to pH 1 and 2. But these bacteria, which is the Helicobacter pylori, is not an acidophile, but how they survive. So what they do, that they surround themselves with a thick layer of polysaccharides, and along with that, they do have this mucus and do secrete some of the specific alkali solution. They just release them from the cell so that it can negate the effect of the acidity in the environment. So the microbial growth has a characteristic temperature dependence with a distinct cardinal temperature. Although the shape of the temperature dependent curves varies, the temperature which is optimum is closer to the maximum than the minimum. This is the typical temperature tolerant curve of the bacteria or the archaea. So here we are looking at some of the microorganisms which are placed in different classes based on their temperature ranges for growth. They are ranked in order of increasing growth temperature, such as the psychrophiles, then psychrotolerance, then you have mesophiles, then you have thermophiles, and then you have hyperthermophiles. And you can easily see that the temperature range or their optimum temperature for a hyperthermophile is close to the boiling water temperature, 95 degrees approximately. These are found in the boiling hot water springs or the hydrothermal vents in the ocean. These thermophiles, on the other hand, can be found also in the hot water springs, which are a little bit cooler and very extremely hot desert as well. Mesophiles are the main organisms that most of the pathogens belong to. The reason, if you look at the temperature, optimum temperature for them to grow, this is around 37 degrees Celsius, so approximately 35 to 40, which is the human body temperature. So most of the pathogens are from the mesophiles, whereas this psychrotolerance can grow at a cooler temperature, whereas the psychrophiles absolutely need cold temperature to grow. Beyond 20 degrees Celsius, they cannot survive either. So here are some of the cardinal temperature for select microorganisms. You can see the cardinal temperature of many of these bacteria, optimum temperatures from 25 to even some of them at 106. So you can easily see that hyperthermophile, that how interesting they are. If you compare to that, to the protists or the eukaryotes or the fungi, there is none beyond this optimum temperature or maximum temperature beyond 47 or 50, 58 degrees Celsius. So beyond that, nothing can survive. Whereas you can see that this Pilobus fumare can survive at the temperature even at 113 degrees centigrade, which is basically steam. So the adaptation of the thermophiles, how they do it, the protein structure is stabilized variety of means. For example, there are more hydrogen bonds to stabilize them. There are more proline. For an example, there are chaperones. So why there are more hydrogen bonds? Many of the proteins have extensive beta sheets. So remember that the alpha helices have lesser hydrogen bonds, whereas the beta sheets are much more stable and more hydrogen bonds. So many of their proteins have evolved to have this beta sheet structure. On the other hand, the proline is a very interesting amino acid where they do have a cis-trans form specific to their CN bond. So they are very rigid on the proteins, and they do give the steric hindrance of a protein that how they will properly fold. And this proline structure will not allow the protein to fold or denature in a certain way, and that can also help their stability at higher temperature. And the chaperone proteins are the proteins which are the helper proteins, which also protect the proteins from them to get unfolded or denatured at higher temperature. Now, the membrane is stabilized by a variety of means. For example, there are more saturated, more branched, and higher molecular weight lipids. And there are also ether linkages in the archaeal membranes. And what this does, if you remember that, you have these phospholipid bilayers in the plasma membrane, where you have a phosphate head group and two fatty acid tails at each end of each leaflet of the plasma membrane, where we talked about that the archaeal plasma membrane is almost a monolayer, where one of these ether linkage groups with the two fatty acid tails are covalently attached with the fatty acid tail of the other one. So you have one cyclic molecule with two polar head groups in each end, connected by these ether linkages to this fatty acid tails. So being just a single molecule gives an extraordinary rigidity or stability at higher temperature for these archaeal membranes. So the next we are going to consider the oxygen concentration, and growth in presence of different oxygen concentration depends on the microbe's metabolic process, electron transport chain, electron acceptor. These are some of the common things. Just trying to give just a basic definition that aerobes, sometimes we call them obligate aerobes, grows in presence of atmospheric oxygen, which is about 20% of O2 of the total. On the other hand, that obligate aerobe requires absolutely oxygen. Without obligate means that without oxygen, they will not survive. On the other hand, anaerobe grows in the absence of oxygen. Obligate anaerobe usually killed in presence of oxygen. They cannot absolutely grow in presence of oxygen. That's very important. Microaerophile grows at a 2 to 10% oxygen, which is lower than the atmospheric 20%. So these microaerophiles are often found in the lake, river, water bodies where they have less oxygen because the dissolved oxygen amount is less, and they perfectly grow as microaerophile in those conditions. Facultative anaerobe do not require O2 but grow better in its presence. So these are the best of the two worlds. Giving a classic example of facultative anaerobe is E. coli. So for an example, when E. coli is in our intestine, which is a colon or the large intestine, lots of E. coli in there, and they are beneficial. They are natural flora, gives us vitamin, but they get our also help in the digestion and eat the food that is undigested in our gastrointestinal tract. So in the gastrointestinal tract, particularly in the colon, there is no oxygen in there. So these bacteria grow in an anaerobic condition, and that's one of the reasons that they are in check. They cannot overgrow because there is no oxygen. But if you are growing E. coli in the laboratory on a flask or anything, they can utilize the O2 and they grow exponentially. They grow extremely fast. And this is one of the adaptation that they have, that they can utilize oxygen if present, but they will not utilize oxygen if it is absent. Aerotolerant anaerobes, on the other hand, grow with or without oxygen. So there is a subtle difference in between that. So just make sure that you understand this clearly. Aerotolerant anaerobe will grow in absence of oxygen, will not utilize it. They will grow in presence of oxygen as well, but still will not utilize the oxygen for their metabolism. They will grow in absence or presence of oxygen, but will not utilize oxygen under any circumstances, even if they are growing in oxygen atmosphere. Whereas a facultative anaerobe can grow without oxygen, but when oxygen is present, they are going to utilize that oxygen for their growth. So that's the basic difference, that facultative anaerobes will use oxygen to grow, whereas aerotolerant do not require oxygen no matter what. So here, what we are looking at, that each of these dots, that what we are seeing is an individual bacterial colony right here. And say, for an example, this could be either agar or this could be a liquid culture. And you have a screw-capped structure at that time. The way we do this experiment, if we are doing this experiment in a liquid culture, we use a broth which is added with a particular chemical which is known as a thioglycolate, or sometimes you can use cysteine as well. So what the thioglycolate does, the thioglycolate will scavenge all the oxygen that is present in the media. But just above the screw cap where you have a little bit of oxygen, that will diffuse to the interface, and we call this top area as the oxic zone. So you inoculate the bacteria, close the screw cap so that no fresh oxygen can flow through, that only the oxygen in that top area is present, and then allow the bacteria to grow, and they will show a growth pattern. And how is this growth pattern? This growth pattern, say for an example, if it is an obligate aerobe, they can only grow at the interface, which is the oxic zone. They cannot grow in the anoxic zone because they absolutely need the requirement, which is the oxygen, for them to grow. On the other hand, see that the microaerophile grows close to the oxic zone, but just a little bit below, because they can tolerate about 2 to 10% of the oxygen in the environment, but not more than that. On the other hand, the facultative aerobe, you can see that they can grow, the facultative anaerobes, all throughout the culture, but their growth towards the top is a little bit more, because you know that they can make more ATPs in the presence of oxygen, because they can carry out this electron transport chain and making ATP. So that's one of the reasons that they are a little bit higher, but they can grow all over the place. These are facultative anaerobe, and best of both worlds. The next one is an aerotolerant anaerobe. No matter whether they are growing in presence or absence of oxygen, they have an uniform growth all throughout. So there is no effect of oxygen, whether it is present or absent. They are going to grow in both conditions equally. They do not utilize oxygen for their growth. That's why it is not dark at the very top. On the other hand, there is a strict anaerobe, or we call this the same way. We call this as an obligate aerobe. Sometimes we call this as an obligate anaerobe. And this is a very strict anaerobe where it grows towards the bottom. That means that these need absolutely no contact with the oxygen. If it comes in contact with the oxygen, this will eventually die. So this is the very important part. Now, there are some kind of enzyme system present in these bacteria which allows them to tolerate or not tolerate oxygen. And what are those? So, and why it is important. So when a bacteria grows into the oxygen environment, at that time, the oxygen that is getting into the cell, they can form superoxide radicals, which are known as an O2 minus. They are known as a free radical, reactive oxygen species. There's our O2 minus, which is with an unpaired electron. Remember that your O2 is perfectly all right. But when this forms as an O2 minus, and this is constantly formed for all the aerobic organisms which use the electron transport chain. At the end of the electron transport chain, these extra electrons are accepted by the oxygen, And for a short period of time, they are going to form these O2 minuses. And these O2 minuses are going to get converted into hydrogen peroxide. And from hydrogen peroxide, they are going to be depleted into the water, molecular oxygen, or just water. For a short period of time, these oxygen receive these unpaired electrons and the O2 minus, and even for that short period of time, immediately these reactive oxygen species will attack the cellular proteins and the lipid to oxidize them. They are extremely reactive and they can denature these proteins and the lipids. So to scavenge these, you have an antioxidant system. We have these. The bacteria also do have this antioxidant system, which scavenge these O2 minus and convert them either to hydrogen peroxide, and from the hydrogen peroxide is toxic as well. So they have to convert the hydrogen peroxide to water and oxygen or just water. To do this, you need usually two enzymes. So the first one is common to all aerobes, which is known as SOD or superoxide dismutase. Everywhere SOD is present, except in the strict anaerobe. As there is no SOD, this will not tolerate any oxygen. But any bacteria which can tolerate oxygen will have the SOD or superoxide dismutase. What it does, it takes that oxygen, which is with the electron O2 minus, and convert them to hydrogen peroxide. That's the function of the SOD or superoxide dismutase. But again, as I said, H2O2 or hydrogen peroxide is still toxic. So you need a second group of enzyme which will convert them to either water and oxygen or just water. So one group of enzyme is known as a catalase. So the catalase will convert the hydrogen peroxide, whereas another one is called the plus minus catalase, low levels. You also do have a catalase. Here you have a without any catalase, without any catalase. So what are the different reasons that you have these catalase? Because catalase will catalyze the reaction to convert the hydrogen peroxide to water. There is another enzyme which is also present in some of the bacteria, Streptococcus, which also do have an enzyme called peroxidase. So this peroxidase, so SOD or superoxide dismutase have to be in all of them except the strict anaerobes, and either they have to have a catalase or peroxidase. And don't worry about that, as long as you remember that you need this antioxidant enzyme system in all of the bacteria that is growing in presence of oxygen that is needed. Only the aerotolerant anaerobe have just only the SOD or superoxide dismutase, but there is no catalase present in there. So the next one we are looking at, the oxygen is reduced, that what we are talking about, to ROS, superoxide radical, hydrogen peroxide, hydroxyl radical. Aerobes produce the protective enzymes, superoxide dismutase, catalase, and peroxidase. So that's what we just talked about. The next one is the pressure, where the microbes that live on the land and water surface live at what atmospheric pressure. Some bacteria and archaea live in deep sea with very high hydrostatic pressure. These are known as the barotolerant, and they are adversely affected in increase in pressure, but not severely as a non-tolerant organism. And there are some which are known as a barophilic or piezophilic organism, required to grow more rapidly in presence of increased pressure. So the change in membrane fatty acid actually is responsible to adapt to this high pressures. These are barotolerant or barophilic. The next one, we are looking at this electromagnetic radiation, and we are constantly looking at the electromagnetic radiation spectrum, in the sense that we are constantly being bombarded with this whole spectrum. Some of them a little bit more, some of them a little bit less, but this is the visible spectrum. Infrared is far red, we cannot see that, and you have these ultraviolets. But along with that, there are some radio waves, there are some X-rays and the gamma rays, and there is a very little amount of these, because these are ionizing radiation and very detrimental to any life. So the radiation behaves as if it is composed of waves like those traveling on the surface of the water. The distance between the two waves crest are basically the wavelength. These are just the basic idea. And the energy of the radiation increases. The gamma rays, X-rays, much more energetic than the visible infrared or the light, but they are detrimental for the cell because they can break the DNAs and cause mutations as well. So the ionizing radiation we talked about just now, that X-rays and the gamma rays cause the mutation that indirectly results in death. This disrupts the chemical structure of many molecules, including the DNA. It causes the breakage in the DNA. And the bacterial endospores, like, say, for an example, Deinococcus radiurans, are extremely resistant to DNA damages. On the other hand, so radiation damage cannot even happen to them. On the other hand, for a high dose result in the death to sterilize items, that is also used. So there are many material that is sterilized by the gamma rays. Classic example that we use these Petri plates made up of plastic in the laboratory for like agar plates or dishes. These are made up of polystyrene. And these, if you look at the package, it is written there that they are sterilized by the gamma radiation at their source. So this is the way that they are being sterilized before packaging into a clean room. So on the other hand, the ultraviolet radiation are the most effectively absorbed DNA at 260 nanometer, and that will cause the mutation. And we already talked about that ultraviolet can cause the thymine to thymine dimers. And these thymine dimers will stop the DNA. There are ways that the bacteria can make the correction to these. Remember that the photolyase was the enzyme which was responsible for excising these thymine dimers out. There are ways, but sometimes still, if that is not controlled, these mutations can lead to the death. The visible light, high intensity, generates the single oxygen. So that singlet oxygen is a powerful oxidizing agent which do have an electron lone pair. This is the same thing. The singlet oxygen is the same thing as the atomic oxygen, and that is also detrimental to cell because it's an extremely powerful oxidizing agent, and it will oxidize lipid and the protein, and that is very harmful. The carotenoid pigment also protect from the light exposed to microorganism from photooxidation. So this helps a lot of microorganisms as well. So these carotenoid pigments are present in the carrots. Beta carotene is one of them, which is the orangish yellowish color. That is your carotenoids. So now we are looking at how the microbes grow in the natural environment. And let's take a look at this, what they do. The microbial environment are complex, constantly changing, and expose a microorganism to overlapping gradients of nutrients and environmental factors. And these contain both micro and macroorganisms. So most microbes inhabit in an oligotrophic environment. What we mean by oligotrophic environment means that there are different trophic levels that we actually just saw. We saw that all these nutritional requirements, and that is we mean by oligotrophic environment, where you have multiple trophics or multiple different types of bacteria or archaea present in one environment. And some microbes live in neutral labile environment where there is a depletion of the nutrients. There is not much nutrient available to them. And also due to this, microbes have evolved many responses to starvation and environmental stress as well. So what are the different things that the microbes can do to live in a growth arrested state if the nutrients are not available? They can change morphological changes like endospore formation. They can enter the stationary phase of the growth curve. They can use cellular component as nutrients. A classic example is that the slime layer forming bacteria, which are producing capsules or slime layers outside the cell, which are made of polysaccharide, which could be used up as a nutrient as well. Very classic example. Numerous genes and proteins also help to fight them through these tough conditions, but not at this culturable state at this point. So these are some of the characteristics of the response to starvation and environmental stress. So here we are looking at the biofilms, and these are very common in nature. So say for an example, most microbes grow attached to a surface. Sessile means they are immobilized in there rather than free floating or planktonic. The advantage is that if they are sessile, they have more access to the nutrients as well. These attached microbes, member of the complex slime enclosed communities called a biofilm. And biofilms are ubiquitous in nature in water. Say, for an example, if you just pick up a rock from a lake or a pond, it feels slimy, and this is your biofilm. And on the other hand, it can be formed on any conditioned surface. What we mean by the conditioned surface. Say, for an example, let's take a look for our tooth. So when you clean the tooth, it is not conditioned. But what happens that in our saliva, there are plenty of proteins, and these proteins coat them or coat our tooth constantly. As soon as the tooth is coated, it is a conditioned surface. On top of that, the bacteria will stick, make colonies, and this will be basically a biofilm. Now, to disrupt that biofilm formation, you can brush your teeth, floss your teeth, and do all of those kind of things, of course, but still biofilm is formed, and plaque is a classic example of biofilm formation on your tooth. So here, what is happening here, the substratum is preconditioning by the ambient molecules. So these needed to be attached onto the surface. Just the classic example that we talked about. Suppose this is a surface of a tooth enamel. So these are some of the protein molecules that needed to be attached on top of that to precondition them. Then you have the cell deposition. Cells are coming in, many different types. They are being deposited. And then you also do have what is called in the third process, right here. You can see the third position is a cell adsorption. They are adsorbed onto this preconditioned molecule. And then they have a desorption. They can go out as well. And then what happens? There are cell-to-cell signaling. So cells can talk in between the different species of the same genus. Sometimes they can talk in between the different genus. Sometimes they can even communicate inter-kingdom communications with plants, bacteria like that as well. So it is a very complicated process. On the other hand, here you are seeing that this is a convective and diffusive transport of oxygen and other nutrients. And then what happens? Replication, growth, secretion of polysaccharide matrix, detachment, erosion, and sloughing is the whole process. So by this process, the biofilm is formed on a surface which is preconditioned, and here the precondition is a protein that you produce to coat them. So the surface of a biofilm is a negatively charged matrix where the positively charged antimicrobials can bind. The biofilm has a nutrient and oxygen gradient, and it's a high concentration on the outer side. So the outside of the biofilm is negatively charged. And there is a low concentration of this negative charge in the middle. So what happens that with a fast-growing species in the outer areas, and there are slow growers in the center. And this figure also identifies the cell-to-cell signals and changes in the cell physiology, persister cells, and the genetic diversity. So there are many different types of growth that actually happens in different part of the cell. So just make sure that you make yourself familiar with some of the terms like the persister cells. So the EPS and the change in the attachment organism physiology to protect the microbes from harmful agent. The organism, when formed on a medical device such as implants, illness can result. So on these implants, prosthetics, or catheters, these biofilms can form, and that can cause illness. Organisms are constantly sloughing, and they can actually contaminate the water phase above the biofilm, such as drinking water system as well. So these are some of the problems that from the biofilm that is being formed in a pipe which is delivering water, and somehow from there they can slough off and they can be contaminate the water system as well. So cell-to-cell communication within the microbial populations is a very interesting area of study, and the bacterial cell in biofilms communicate in a density-dependent manner, which is known as a quorum sensing. And they produce small proteins that increase in a level such as microbes replicate and convert a microbe to a competent state. And in this case, the DNA uptake occurs and the bacteriocins are released. So we are going to talk about this in a second. So essentially, this was an example in Streptococcus pneumoniae. So what happens is that all these bacteria, when they are inside the tissue and replicating, they will produce a condition where they will convert themselves at a certain, they will release a peptide, and at a certain concentration of this peptide, they will become competent cells. Competent cell means that they will be able to take up DNA from the environment. So once all these cells are become competent at that time, the cells will release bacteriocins. And what the bacteriocins will do, that the bacteriocins will kill or destroy all the ones which are not competent yet. That means that they have failed to become competent cells to take up any DNA from the environment. So as these cells are killed or lysed, their DNA is going to come out of the cell. Now, all the rest of the competent cells, which are already competent, are going to take up the DNA from these lysed cells, and these actually can contain the virulent genes and all these different types of genes, and that will make the population, again, to some extent, recombine with different ones, and they will produce these kind of normal recombinants, which is not artificial, but just directed by the environment in their natural conditions. So right here, what we are looking at, that this is the N-acyl homoserine lactone, is known as an autoinducer. And what we are going to look at that this is actually produced by a particular organism, a gram-negative organism, and there are many other. And most of them, if you take a look at them, that they looks pretty hydrophobic. And as they look hydrophobic, they diffuse across the plasma membrane. On the other hand, once inside the cell, these allow the expression of target genes regulating a variety of functions. So let's take a look at how this works. So representative organisms and functions regulated in a Vibrio fishery, like it produces a bioluminescence, Agrobacterium tumefaciens. This causes plasmid transfer. Erwinia carotova, it causes the virulence and antibiotic production. Pseudomonas aeruginosa, virulence and biofilm formation. Then you have a Burkholderia sepia, virulence. Then you have farnesyl borate. These are the representative organism and functions are regulated by this. So basically the overview of these, that how these molecules or the quorum sensing happens. So suppose just giving an example of this N-acyl homoserine lactone. So when the bacteria is inside an organism, or it is replicating at that time in a tissue or anywhere, it is producing these in the cell. And inside the cell, you have a little bit of higher concentration. Outside the cell, you don't have much. So it is diffusing out of the cell, down the concentration gradient, high to low outside the cell. Now there are many cells, all of them are producing these, and they are getting outside the cell. Gradually, when you have a large population that is going to produce a huge amount of these homoserine lactones, and they are all going to go outside into extracellular space, and the amount of this N-acyl homoserine lactone is going to increase at a particular point. When the concentration of the homoserine lactone increase beyond a threshold outside, means that the outside concentration is now more than the inside the cell, this N-acyl homoserine lactone is going to move back from outside the cell to inside the cell. That's the trigger or the switch or the quorum sensing, that at that time, when the homoserine lactone that is going to be moving from outside the cell to the inside of the cell understand there is a good amount of population in the environment or just outside. And at that time, it will produce some particular virulence to do some harm. So the next part we are looking at, that right here, is this warm water squid that remains buried in the sand during the day, and it actually feeds in the night. And right when feeding, it uses its light organ located in the ventral surface to provide this camouflage, projecting light downwards. Thus, this outline of the squid appears as bright on the water surface to potential predators looking up through the water column. The light organ is colonized by a large number of Vibrio fishery cells, and these are autoinducers, which accumulates to a threshold concentration, triggering the light production. So this is a classic example of this quorum senses system. And there are a couple of different ways that the symbiosis, Vibrio fischeri bioluminescence, which helps the squid for predation, pathogenicity and increased virulence factor production, DNA uptake for antibiotic resistant gene, and also there is interdomain communication. What we mean by interdomain, sometimes called interkingdom communication, say for example, rhizobium can interact with these leguminous plants for the nitrogen fixation with flavonoids, NOD-D, or the NOD factors. So there is lots of beneficial effect for quorum sensing systems as well. So the next part, we are going to talk about the laboratory culture of the cellular microbes. And let's take a look at it, what we are going to do. So the culture media is need to grow, transport, store microorganisms in the lab, and the culture media is a solid or liquid, and we're going to talk about a couple of them. So the first thing that what we are talking about is a defined or synthetic. Each ingredients can be defined with a chemical formula. And these are much less used until we are using for a particular type of experiment and labeling experiment. We can use this defined synthetic media, whereas most of the time for regular bacterial growth, we use a complex media, some ingredients of unknown chemical composition. So chemical composition, defined synthetic and complex. Physical nature, liquid, semi-solid, or solid. We can utilize any of those, and they are supportive general purpose and also enriched and also selective and differential. So what we mean by these functions, enriched will allow only a specific type of bacteria to grow in there, and selective means they are, again, will allow only a specific type of bacteria to grow, not others to grow. And differential means that along with the growth, a certain types is going to do a specific type of reaction with the media to give you an idea that how you can identify that organism. So some of the media components in the complex media are the peptones, which are the protein hydrolysates from the protein sources. There are some extract, like in aqueous extracts, usually beef or yeast. Then there are agar, which is a sulfated polysaccharide solidifying agent from some of the seaweeds. And most microorganisms cannot degrade it. And this is a solidification agent. And these are some of the material that is found in your LB agar. So here, on the other hand, we are looking at a functional type of media. So the supportive or general purpose media, for example, is a TSB or TSA, which is a trypticase soy broth or trypticase soy agar. So this trypticase soy broth or agar, this will support the growth of everything. So they are general purpose media. On the other hand, this enriched media, which is, say, for example, a blood agar media, is also supplemented, a general purpose media supplemented with a special nutrients. Means for the blood agar media, the regular nutrient media is supplemented with usually sheep blood. Why it is necessary? Because they can give us some specific type of results or growth of some of the specific type of microorganism. So for an example, this top one is known as a chocolate agar. How it is produced? You have a blood agar plate, which will more look like this, and then you heat that blood or increase the temperature of the media to about 60 degrees Celsius, and the blood is going to get charred to this chocolate, and then it will solidify. Now, once it cools down, you put your bacteria on top of this, and this grows. Say, for example, your book tells that there is a Francisella tularensis, which causes tularemia. It's an extremely virulent disease that grows on chocolate agar media. There's another common bacteria, which is a Neisseria gonorrhoeae, which causes a gonorrhea, that is grown on the chocolate agar media. These bacteria are called fastidious. What do we mean by that? They need a specific type of nutrients which is present in the chocolate agar, which is released from the blood, some of the blood proteins and factors needed for their specific growth. Without that, they will not grow on a regular media like TSA or TSB. So that's your enriched media. On the other hand, there could be functional assays from the media in the blood agar plate. This is a blood agar plate, and there are two colonies. And what you are seeing, that there is a zone or a halo around each growth. So what they did. So the blood agar contains the red blood cells, and these red blood cells are lysed into this area and it cleared that area. So it lysed the cells. That's why it looks clear or yellowish. So this bacterial colonies produce a particular type of chemical, which we call as a hemolysin. Hemolysin means that it can break down the red blood cells or destroy them and gives a very characteristic growth pattern or in there to identify that. Say, for an example, all the staphylococcus produce this hemolysin, so they will produce these zones like this. So the selective and the differential media. A selective media allows the growth of a particular microorganism while inhibiting the growth of others. So let's first take a look at this McConkey agar, which selects the gram-negative bacteria. What do you mean by this? If you put any gram-positive bacteria on a McConkey agar plate, it will not grow. It contains crystal violet in there, which binds to the gram-positive organisms on their peptidoglycan, doesn't allow them to grow. Anyway, so that's not needed. But only the gram-negative bacteria will grow on this McConkey plate. So it's selecting the gram-negative bacteria. This is also a differential media, and we'll talk about that differential function right here in a second. On the other hand, so the differential media distinguish among the different groups of microbes and even permit the tentative ID based on their biological characters. Say, for an example, a blood agar, we talked about that just now, distinguish between the hemolytic versus non-hemolytic bacteria. What do you mean by the non-hemolytic or hemolytic? Hemolytic means that they produce the hemolysins. You have this particular halo type structure around the colony. If you don't have that, that means it is a non-hemolytic bacteria. That's the differential distinguishing. McConkey agar, on the other hand, distinguish between the lactose fermenters versus non-fermenters. So we already talked about that the McConkey agar selects gram-negative. So all the bacteria which grows on the McConkey agar are gram-negative. No gram-positive will grow. But among the gram-negative bacteria, we can tell which gram-negative bacteria can ferment lactose and which gram-negative bacteria cannot ferment lactose. So if the gram-negative bacteria cannot ferment lactose, they are going to grow as a tan colony on the McConkey agar plate, a tan colony. On the other hand, if it can ferment lactose or the gram-negative bacteria can ferment lactose, it's going to produce a red or a pink color. And the way it works that you have this, the lactose fermentation will cause acid production, and that will drop the pH of the media, and dropping the pH of the media will cause a colored reaction to produce that pink color. So you can see that the McConkey agar is a selective and differential media both. And it is actually true that most of the media that we use routinely in the laboratory, that they are really for two different purposes. They either are selective, differential, both type of media, so that we can easily do multiple studies through these plates. Now, here we are looking at a system which is known as a strict anaerobic methods. And all strict anaerobic microorganisms lack or have a very low quantities of superoxide dismutase and catalase. And these microbes cannot tolerate O2 and must be grown without O2. If they do not have any superoxide dismutase or catalase, they will not be able to tolerate oxygen even for the slightest time. If they can tolerate it just for a little while, they do have a very low quality. So you have to handle them in a chamber like this, which is completely enclosed. You used your gloved hand to inside, and this will be completely under vacuum. There is no oxygen in that atmosphere. You have to put them and put it in another chamber, and you do everything in this anaerobic chamber. On the other hand, how you grow easily, this is the manipulation, and this is only required if the strict anaerobes cannot even tolerate the minimal exposure to oxygen. On the other hand, in the laboratory, most of the strict anaerobes, they can just tolerate the oxygen the time it is needed to transfer from one plate or tube to the other. This just takes a couple of seconds to a minute, and that short exposure should not kill them. But how we grow them? We put them in a system which we known as a gas pack jar. It's known as a gas pack jar, and what we have here, we have a catalytic sash in here, which we open up and put it outside this plates. So we put the plates in here, we put an indicator, which is a methylene blue, and we put this gas pack inside. The envelope basically generate hydrogen and carbon dioxide, and that will scavenge all the oxygen from this chamber. And after we open this gas pack envelope and put it in there, just within about 20-30 minutes, the whole atmosphere in here is going to be completely anaerobic. And how do you know that it is anaerobic? You have a methylene indicator strip. Methylene blue becomes colorless in absence of oxygen, whereas it's going to be blue in presence of oxygen. So when you put this methylene strip inside that gas pack chamber, it's going to be blue because it's aerobic. Just within about 20-30 minutes, it's going to turn in white. That means that your experiment is working fine. And here on those plates, these are some of the strict anaerobe that can be growing. We are looking at the isolation of the pure cultures and the population of cells arising from a single cell developed by Robert Koch. And he allowed the study of single type of microorganism in a mixed culture with a spread plate, strict plate, pour plate technique to isolate these pure cultures. So let's take a look at that. These are pretty easy. So this involves the streak plate technique, a spreading mixture of the cells on the agar surface so that individual cells are well separated from each other. So each cell can reproduce to form a separate colony, visible growth, or clustered microorganism. So what we do that we take our loop that we use in the laboratory and we take our inoculum of the bacteria that we want to grow, and we go into this first quadrant and just make a couple of streaks. Then we flame our loop so that it is completely sterilized, and we touch this second area and we streak a couple of times into the second area of the quadrant. So the purpose is that we are diluting this bacteria into the second area. Then we again flame our loop. We go from the second area to the third area. And then finally that what we do from that third area, we just go in the middle. So you can see that first, then you have right here, this was basically the first, second, and then third. As we are trying to do these different types of movements, first, then you have the second, then you have the third, then you have the fourth. These are the individual colonies from a streak plate, and you can isolate a single colony that is grown from just one cell. You can, of course, argue if there are two cells sticking together, can you distinguish? Of course you're not, but for all practical purposes, one colony is arising from one bacteria. The next one is a spread plate technique, and it's usually a small volume of diluted mixture containing about 25 to 250 cells are transferred, and they are transferred to a plate, and then you have a glass spreader which is like this, shaped like this, and then you just go back and forth all over the place. Just make sure that you spread all these bacteria and you will see individual colonies. And then you have a pour plate. So this sample is initially serially diluted. Why this experiment needed to be done? Say, for example, you are growing some kind of bacteria. If you are putting those bacteria onto the plate, you have millions of bacteria you will not be able to count, because remember that you have to have something in between 25 to 250 cells. Other than that, it is almost impossible to count. If you just even take 0.1 or 1 ml to there, you are probably transferring millions of cells in that volume, and you cannot count it. So what you do that you take your original culture and you serially dilute it tenfold each time. So each of them contain about 9 ml of water or saline, usually most of the times 0.9% sodium chloride. And then from the first one, you take 1 ml of your sample to 9 ml. So now it becomes 10 ml. So this is your 1/10 dilution, or 10 to the power -1 dilution. Then again, you take 1 ml from here, put right here. Now this becomes another 1/10 dilution. And if you now do 1/10 into 1/10, you multiply that, it becomes 100th or 10 to the power -2 dilution, 10 to the power -3 dilution, 10 to the power -4 dilution, and so on you go on. So now you are going to go to some of the like a 10 to the power 5, 6, 7 dilutions, or even 10 to the power 4. And you plate these bacteria and try to count. You have to do this trial and error to see that where you have the colony number in between this 25 and a 250 cells, which is a kind of a rule that is the optimal number of colonies to be counted. If you have too many colonies on one plate, you cannot count it. And if it is too less, it is statistically not significant. So you like to have about 25 to 250 colonies per plate. So this is your pour plate technique. Now we are looking at that the growth of the microbial growth on the solid media, and there are colony characteristics that we can note. And when we grow these can be noted to look at all these different types. And if we just look at that, what we are looking at, these are the form. Then we're looking at the elevation. Then we are looking at the margin. And these help in identifying this microorganism by their type, and these helps. And the differences in the growth rate from the edges to the center is due to the oxygen, nutrients, toxic products, and cells may be dead in some areas as well. So let's take a look at these, that some of these terminology that what we have is this, the punctiform is a small circular dot of varying sizes. Then you have this punctiform. Then you have the circular right here. Then you have a filamentous. Then you have an irregular rhizoid with these fine structures. And then you have this spindle shape. On the other hand, if you're looking at the elevation, you have a flat, raised, convex. Then you have this pulvinate, then umbionate where the middle portion grows up. These are some of the elevations. On the other hand, here you can see that is entire, whereas you can have an undulate, like wavy form, lobulous right here. Then you have an erose, like really jagged lines. Then you have this filamentous as well, and also curve. So there are many different types of morphology that you can actually see. When you are growing some of these microbes, you can note these colony characteristic down. And of course, there will be different many colors as well. So the next part, we are going to look at the microbial population size can be measured directly or indirectly. So let's take a look at this, that how we can do this. So the measurement of the microbial growth can measure the changes in the cells in a population. And we also can look at the mass of the population. And the direct measurements is by counting chambers on membrane filters, flow cytometry, or electronic counters like a Coulter counter. So let's take a look at the counting chambers. So this is a very easy, inexpensive, and relatively quick. And this needs a slide, which is a special type of slide, which is known as a hemocytometer. This is routinely used by the pathologist to count your blood cells, red blood cells, white blood cells, after staining them. This can also count the bacteria, a little bit more difficult, but one of the drawbacks is that it can count the total bacteria, but cannot distinguish the living from the dead cells. That's one of the drawbacks of the counting chambers. So there is also another process which uses a direct count on the membrane filters. So these membrane filters are 0.2 micron filters with a dark background, and it only allows the other things to pass, or the liquid media to pass through, and all the bacteria will be on top of the membrane. And then you stain them with a fluorescent dye and you look at them and you can count the bacteria. With certain dyes, you can also distinguish living or dead cells, because certain fluorescent dyes will be taken up only by the living cell, but not by the dead cells. So this is another quick count on the membrane filters, direct individual count of the bacteria. The flow cytometer or cytometry is a price technique by which you can use a microbial suspension. This is forced through a small orifice or a hole, which does have a laser light beam. And when these single stream of microbes through this orifice moves, it scatters light, or the laser. And each light scattering event is detected independently, and this number of events represent the number of cells. And the cells differing in size, internal complexity, and other characteristics within the population can also be counted. So what you can do, that you can label a certain type of cell with a certain type of fluorescent dye with multicolors depending upon cell type, and you can use those to even separate them with the flow cytometry, and also you can count them. Say for an example, I'm just giving an idea that if you're trying to identify some of the T cells, you isolate it from a spleen, like a spleen tissue. So what you can do that you can label these cells, particular antibodies, against a cell surface protein. So these T cells will have a T cell surface receptor, and these T cell surface receptors will bind to a specific antibody that you have raised or commercially available, which is bound to a fluorophore or a fluorescent dye. Now, you can attach that fluorophore or the fluorescent tagged antibody to that particular receptor, and you can just easily identify that how many T cells are in the population of those spleen cells, or even by some sophisticated technique that which is called the FACS, which is a little bit advanced than a flow cytometer just by itself. Flow cytometer just counts, whereas the FACS or fluorescence activated cell sorter use the same mechanism, but depending upon a tag or a particular color, it can sort the cells or isolate the cells as well, along with the counting. So the electronic counter, so the Coulter counter, the microbial suspension is forced through a small hole, same thing, and electric current flows through the hole, and the electrode placed on both sides of the hole measures the electrical resistance. Every time a microbial cell passes through the hole, the electric resistance changes. For example, the conductivity drops and the cell is counted. So this is your Coulter counter. So this figure describes the states that a cell can exist in between alive and dead. Actively metabolizing cells, cells with reduced metabolic activity. There are some cells with metabolic activity and plasma membrane intact, but RNA content is reduced. Plasma membrane intact, but no detectable metabolic activity. Extensive damage to the plasma membrane, cellular DNA degraded, and then cell fragments, and they are dead. So there are many different conditions that a microbe can be in before they can be considered as actually dead. The next type of technique is a viable counting methods, and it's a standard plate count. And your spread and pour plate technique that we talked about can easily do this. So your diluted sample of bacteria is spread over the solid agar media surface, and after incubation, the organisms are determined by counting the number of colonies. So each one of them is expressed as a colony forming unit. Now remember that when you are counting the colonies from your spread or pour plate technique, one colony is arising from one bacteria. So you can easily calculate that if there is 20 bacteria on one plate or 20 colonies on one plate, those 20 colonies means there were initially 20 bacteria plated onto that plate. So you can easily look at the dilution of that. So you can just go back, say for an example, here you have this 1, 2, 3, 4, 5. So you can see that from here, you just put 1 ml right here, 1 ml of this into this plate. So 1 ml of this 10 to the power negative 4 contains 1, 2, 3, 4, 5 colonies. So you can calculate that if the 10 to the power negative 4 contains 5 colonies in 1 ml, how many bacteria will be there in the original culture. You can easily calculate that with a simple formula. So here on the other hand is a viable counting methods by a membrane filter method. So the bacteria from the aquatic samples are trapped onto the membrane. Membrane is soaked in the culture media and the colonies grow on the membrane. And these colony counts determine the number of the bacteria in the sample very rapidly. So here what we do. So here is a setup of a filtration system where you have this 0.2 micron bacteriological filter. You take your environmental sample, pass through it with a vacuum. So it will pass the liquid and it will trap all the bacteria on top of the membrane, because this is a 0.2 micron membrane. That means at 0.2 micron pore size, bacteria will not be able to pass. They will be stuck on there. Then you just touch that onto nutrient media and you allow it to grow. Sometimes these membranes are also transferred onto the Petri plate, or you can just grow by itself. So now you can count the number of colonies on that particular membrane, and that will give you the numbers in that particular sample. So this is, you can easily back calculate. So this is viable counting method. The reason that this is very, very good, because each colony arises from each live bacteria. So this will not count any dead bacteria. On the other hand, there is also a viable counting method which is measured by the most probable number, or the MPN method. And these are where the microbe cannot be cultured on solid media or will overgrow the surface of the plate. The dilutions are made and added to a suitable media, and there is a turbidity measurement to yield the most probable number, or NPN. How do you do this? So let's take a look at these methods. One is a dry weight. Time consuming, not very sensitive. You can just collect as a cell pellet by centrifugation. You can weight that. And before you do that, you have to dehydrate the bacteria so that there is no water mass is present in there. Then you can take the dry. Spectrophotometry is a classic example about light scattering, is directly proportional to the cell biomass. So what you do is, like, you have the bacteria and you shine a monochromatic light on top of that. And depending upon the concentration of the bacteria in the solution, it's going to scatter light, or it will scatter less or more. What do you mean by that? A dilute solution will scatter the light less because there are less particles. A concentrated solution will scatter the light more because there are more particles or bacteria present in there. So this is collected automatically through a detector, and computationally you can just plot a graph beforehand, and you can just fit those data into your graph to find the number of cells in that particular sample. On the other hand, the concentration of a particular cell constituent, for example, you can measure the amount of DNA, ATP, or chlorophyll, and useful if substance in a cell is constant, you can easily measure these concentration of the cell constituents. So the next part, the last part, we are going to talk about the chemostat and the turbidostats are used for continuous culture of microorganisms. Because we already talked about that the closed system cultures, like a flask or a test tube, once the culture media get depleted and there is deposition of the toxic metabolites, the bacteria is going to first show a flat stationary curve, and then they're going to go into a death phase or declination phase. To avoid that, we can use a chemostat or turbidostat, and you can think about that. These are also termed as fermenters. Sometimes these fermenters are used in the laboratory, or sometimes there are very sophisticated fermenters that are used in the biotechnology or the pharmaceutical industries to produce a lot of different things. So the continuous culture is an open system, continual provision of nutrients, continual removal of a waste that we talked about. This maintains cells in a long phase at a constant biomass concentration for extended periods. And this is achieved using a continuous culture system where you have a continuous supply of cells in exponential phase grown at a known rate. Now, the study of microbial growth at a very low nutrient concentration, close to those present in the natural environment, study of interactions of the microbes under conditions resembling those in aquatic environments, and also a lot of experiments can be done for the food and the industrial microbiological settings. So here we are looking at a chemostat, the rate of incoming media or rate of removal of the medium from the vessel, and essential nutrients in the limiting quantities, most stable and effective at lower dilution rates. So you can easily see that there are fresh media, and here you have the media being removed from, and you have the control valve, air outlet, air supply. Turbidostat regulates the flow rate media through a vessel to maintain a predetermined turbidity of the cell density. And the dilutions rate varies, contain all nutrients in excess, and operates best at high dilution rates. So chemostat and turbidostat, just know that what are the different advantages there. Chemostat is advantageous at a lower dilution rate, whereas turbidostat is operated at best high dilution rates. So those are the advantages of each system. So here we are looking at the dilution rate and the microbial growth. So dilution rate at which the medium flows through the vessel, and know the cell density is maintained at wide range of dilution rates. And the chemostat operates best at low dilution. So you can see that this is the dilution rate right here, and this is the generation time. So here, the overview is that the both population size and generation time are related to the dilution rate. When dilution rate are very, very low, there is only a very limited supply of nutrient is available, and the microbes can conserve only a limited amount of energy. And much of this energy must be used for the cell maintenance, not for growth or reproduction. Slightly higher dilution rate makes more nutrients available to the microbes. When the dilution rate provides enough nutrients for both maintenance and reproduction, so you can see that cell density will begin to rise. In other words, the growth rate can increase and the generation time can decrease when the total available energy provided by the nutrient supply exceeds the maintenance energy. For a wide range of dilution rates is almost completely depleted by the rapid reproduction rate of the cells, as seen when there is a decreasing generation time and the cell density is decreasing. At this point, this is because the limiting nutrient is almost completely depleted by the rapid reproduction of the cells, and this dilution rate is too high, and the microorganisms can actually be washed out of the culture vessel before reproducing. The dilute rate is greater than the maximum growth rate. That's pretty much this chapter on growth, and understanding how microbes grow is an important in controlling and encouraging that growth of other purposes. The last part of the chapter is a lot of laboratory-based experiment, and most of these experiments we actually do carry out in the laboratories, and these can give you guys some ideas that if you want to set up some of these laboratory experiments in the laboratories, and that will be really helpful. The last part of the chapter are really helpful to have some application-based purposes. So that will be the end of chapter 7.